And you are welcome, all of you, to ask question after the talk via chat or asking to ask directly your question. Voilà, go ahead, Shensi.
Thank you very much. So thank you very much for the invitation, and it's really a great pleasure that I can share some of our work that I have done in France, and also that continue to do the project in China now. So my work is focusing on the melanoma treatment. So I think most of the project we're doing, it's not so close to the immunology, but I hope that some of these topics will stimulate some thinking in the domain of the onco-immunology. So now I work in West China Hospital in the west of China in Chengdu city. And also I still have some very close collaboration with the Gustave Institute in Paris. So as you may know that melanoma, especially for those people working on onco-immunology, it's one of the most aggressive skin cancer. And the melanoma harbors like 50 to 60% of the BRAF mutation, which is a activator in the MAP kinase pathway. And but 20% of this type of skin cancer, they also harbor the NRAS mutation. And the rest of the melanoma harbors some other mutation like NF1 and also P10. So during last decade actually, before the 2011, in fact, the melanoma, we do not have any treatment because since it's a skin cancer that induced by the UV exposure, so in general, they do not respond very well to the chemotherapy or radiotherapy. But since 2011, two strategies has been emerged for the treatment of melanoma. At the one part is the target therapy targeting the BRAF mutation, as I mentioned before. It's called the vemurafenib. And at the same time, in fact, it was also developed for the immunotherapy with the anti-CTLA-4 inhibitor ipilimumab. And since then, the response rate of the melanoma has been increased a lot compared before the 2011, especially for the target therapy. More than 80% of the response rate in the melanoma patient has been observed with the monotherapy of vemurafenib. But on the other hand, for the immunotherapy, ipilimumab, it was almost to 10% of response rate at that time. And during this decade, there are some other target therapy for the BRAF MAP kinase pathway inhibitors has been developed, especially with the combination therapy of dabrafenib plus the trametinib has been approved in the course of 2014. And on the immunotherapy part, in fact, since the discovery of the PD-1 proteins and development of anti-PD-1 antibodies, two main anti-PD-1 inhibitors was approved for the treatment of melanoma as the first-line treatment. And since 2019, in fact, there are another two new target therapy, oncorafenib and binimetinib was approved for the first-line treatment of BRAF mutated melanoma patients. And at the same time, the combination of the target therapy and immunotherapy, especially anti-PD-1 therapy, has been developed for the treatment of metastatic melanoma. So as we can see from this slide, in fact, now we have the clinical trial data to compare all these kind of different type of therapy for the metastatic melanoma compared to chemotherapy, which the melanoma does not respond very well. The BRAF inhibitors, especially BRAF plus MEK inhibitors combinatorial treatment has a very high response rate at the beginning of the treatment, but the patient relapsed very fast, almost after 1 year. But for the immunotherapy anti-CTLA-4 inhibitor and also anti-PD-1 inhibitors, in fact, we can see that at the beginning, the response rate is not very high. With the combination of CTLA-4 and the PD-1 inhibitors, we have almost like 40% of the response rate in metastatic melanoma patients. But the advantage of this immunotherapy is that if the patient respond to the immunotherapy, the long-term beneficial will be observed compared to the target therapy, which will relapse within 1 year. And now we have another clinical trial that we have also already followed the patient more than 7 to 10 years. And some of the 20% of the patient actually they are still responding to the immunotherapy, and the 30% of the patient they has already stopped the anti-PD-1 or anti-CTLA-4 treatment, and they are still respond to this. The immune system is still working for the melanoma. So the problem for us, especially for the basic research, is that from the clinical observations that actually we arrived at a plateau that there are still like more than 30 to 40% of melanoma patients, they do not respond to any of these kind of treatments. And we're arriving at a ceiling glass effect that we have to overcome in the future. I think it's the most important goal for most of the researchers working on this type of cancer. So if we compare the two first line treatment for the metastatic melanoma, for the target therapy, we can clearly see that they have a very high response rate, but this response is not very durable and they relapse within 1 year. But for the immunotherapy, the problem is the primary resistance. We have very low response rate, but when the patient responds, they have a very durable response to the anti-PD-1 immunotherapy. So from the clinical point of view, actually, we have this kind of RECIST evaluation for the response of the patient for the treatment. So as we know that if one patient have a radiological complete response, actually doesn't mean that there are no tumor cells in the human body. So from this slide, as you can see that for the target therapy, we have a very fast response. That this is spectacular for this patient that after 2 weeks of treatment of target therapy, all the metastatic lymph node metastatic of melanoma has disappeared. But the problem is the 1 year after, all of them come back. And also we can observe that most of the resistant tumor cells actually were stay at the same site compared to original tumor. So what we were thinking is this kind of what we called MRD, the minimal residue disease, that we cannot detect from the CT scan, usually using in the clinics, which represent a subpopulation of tumor cells maybe hiding after the treatment in the human body, and they can relapse after several years. So we come up with this concept that we call it a persistent cancer cells, meaning that the original tumor cells, the parental cells, when we are treated with the target therapy or immunotherapy, some of the subpopulation of these cancer cells, they can switch their phenotype into a persistent cancer cell that they can survive this treatment at the beginning of the therapy. But if we withdraw this kind of target therapy, these population of cells, they can reverse back to the original phenotype that they are sensitive again to the same treatment. But if we keep this target therapy for long-term treatment, so these kind of persistent cancer cells, they have the capability to further evolve into secondary genetic mutational resistance. That is a cell state is stable, that they cannot reverse anymore back to the parental original cell types. And then we will observe the relapse in the patients. So we were trying to understand this population of cells that we could actually is kind of like adaptive resistance, because their it's kind of a switch of phenotype. They do not harbor secondary mutations compared to original populations. And when we remove the drugs in vitro, for example, as I shown here, their sensitivity to the target therapy was very resistant to the target therapy at the day one when we recovered those survival cells from the petri dish. But when we remove the target therapy after several days, these cells, they can reverse back to original population and they have very similar sensitivity to the same treatment. So, which means that this kind of phenotype is transient, adaptive, and also reversible. So to understand the underlying biological mechanisms, we were mainly focusing on the translational level of those proteins involved in the survival of this persistent population of cancer cells. So we're using a technique called polysome profiling, as I shown here. In fact, when within the cells, when the mRNA they need to translate into a protein, they need to be scanned by the ribosomes in the tumor cells. So with these technologies, we use the sucrose density to separate different translation activity of mRNAs based on their binding with the number of ribosomes. So when the mRNA is highly translated, they will be bound by several ribosomes, so their mass is much heavier. So when we are centrifuge this kind of sucrose density, we can separate the mRNAs based on their translation activity. So finally, the profile we're showing here on the left actually is those mRNAs are not bound by the ribosomes, so we call free mRNAs. They are not translated into proteins. But with the increasing density of the sucrose, as we can see here, all the peaks with a higher bonding of the polyribosomes, showing that these part of mRNAs are highly translated in the cells. So with this technique, we were following during the several days of the persistent cancer cells without the treatment. And we have found that actually at the day one, when we recover the survival cells compared to the parental cells, in fact, there is a global inhibition of translation activity, as we can see here with the blue line. But during the culture without the drugs, in fact, as we saw before, there is a reversible effect on the sensitivity to the drugs. At the same time, we can observe here actually the translation activity is also reversed to the similar level compared to the parental cells. However, if we keep the drugs here for the green line, as we can see here, after 9 days of culture, actually the translation activity is still inhibited in the persistent cancer cells cultured with the target therapy. So it means that this continuous treatment of target therapy can keep these cells within this phenotype, and maybe they can further evolve into a secondary mutational genetic resistance. So we have found, based on these techniques, we did some RNA sequencing and we identified like more than 200 mRNAs actually. Even with the global reduction of the global translation activity, these 200 mRNAs actually they are upregulated at the translational level in the persistent cancer cells. As we can see here, some of the candidates, for example, the CBP protein, the mTOR complex subunit proteins like Richter, are actually upregulated in the persistent cancer cells, but their protein expression level were reversed after 9 days withdrawal of the same treatment. But if we keep the treatment, of course, the protein expression were kept at a similar level compared to the day one of the persistent cancer cells. So as we can see here, actually, from several point of view, that those persistent cancer cells have this reversible phenotype for the sensitivity to the treatment and reversible translational modulation, and also the reversible growth rate for the persistent cancer cells. So by using a single cell RNA sequencing, we were trying to follow how these cells can evolve during the treatment. So with the previous data set, actually we reanalyzed the data set that published in 2018. They were using a PDX model of the melanoma to try to follow these single cell persistent cancer cells in the mouse. And in this study, we have shown that, in fact, the persistent cancer cells, they still have some kind of micro heterogeneity within this population. So in general, for the melanoma, we can separate them into 5 different cell states based on their gene expression signatures. And by using this pseudo-time algorithm, we can see that at the beginning of the treatment, most of the cells, they are proliferative, but in the course of the treatment, some of these cells can further evolve into a SMC state, which means a starvation and like melanoma cell state. And then at a branch point, these SMC cell states, they can further differentiate into different phenotype. At one hand, they can differentiate in the pigmented, which is MITF highly positive melanoma cells, and on the other hand, they can differentiate into less differentiated melanoma cell state, which is very similar to the neurocrest stem cell-like melanoma cells. So based on this single cell trajectory analysis, we further looked into the expression signatures of the different cell states, and we were highly interested in for this SMC cell state, since this cell state looks like a original the population, the first survival cells that they can further evolve into different phenotype. So we were analyzing their gene signature and found that there is one very interesting pathway, fatty acid oxidation, was upregulated in this subpopulation of persistent cancer cells. And so we were trying to understand whether this kind of change the metabolism in the persistent cancer cells can promote their survival to the initial treatment of the target therapy. So we used one strategy that by in vitro, by culturing the persistent cancer cell melanoma cells with the fatty acid probes and the biopsy C16 to follow their fate within the cells. As you can see here, actually for the parental cells and the persistent cancer cells, they have very similar uptake activity of the fatty acid from the extracellular medium. But the difference between them is that for the persistent cancer cells, they have a very high consumption rate of the fatty acid they have uptake from the extracellular matrix. Means that they have a higher capability to utilize this kind of nutrient to promote their survival. So to consolidate these observations, we further have done a metabolome fluxomic analysis to follow the carbon-13 labeled palmitic acid to see whether they have a different activity integration to the TCA cycle within the persistent cancer cells. As you can appreciate here, actually for the persister cells, we have effectively higher integration of the carbon-13 labeled palmitic acid integration into several intermediate metabolites of the TCA cycle of the cells, for example, malic acid and the citric acid. And with fatty acid, oxidation inhibitor, the ethyluridazine, we found that actually we can inhibit this consumption of the palmitic acid within the persistent cancer cells, which suggests that this inhibition of the fatty acid oxidation pathway may preferentially kill the persistent cancer cells, which is resistant to our first-line target therapy treatment. On the other hand, it was intriguing that, as I told you, that the global reduction of the translation in the persistent cancer cells, under this background, we still find like 200 mRNAs are upregulated at translational level. So we were wondering whether they have different features of these mRNAs compared to the other mRNAs that globally reduced at the translational level. So there's one mechanism that we were interested in is the post-transcriptional modulation of the mRNA. And as you can, as I shown here, the mRNA, when they were transcribed from the DNA, actually at the same time they were modified, methylated at different side of the mRNA body. And these methylated mRNA, they will be further exported into the cytoplasm, and then they can be recognized by different readers of the methylation on the mRNA, and then launch the ribosome scanning for the translation. These methylation of the mRNA, they were shown to be involved in the translation regulation, and also the stability of the mRNA in the cytoplasm. So we're wondering whether the methylation of this subpopulation of mRNA that we have identified in persistent cancer cells were involved in the regulation of their translation activity and also the survival of the cells. So to answer this question, actually we come back to do the polysome profiling, and we isolated the mRNAs that are highly translated in the polysome fractions, both from the parental and the persistent cancer cells. And we extracted the mRNA, and we did several methods to detect the methylation of mRNA M6A by dot plot, and also the mass spectrometry, and also by M6A RNA sequencing. And what we have shown here is that actually, indeed, the mRNAs that are upregulated at translational level in the persistent cancer cells actually harbor higher 5' untranslated region mRNA methylation compared to those mRNAs in the parental cells that are highly translated. So it means that there is a selection of mRNA translation activity based on their post-transcriptional modifications. And the main enzymes that are responsible for the methylation of the mRNA, the METTL3 and the WTAP, which was in the same complex of the methylation enzymes. And we have shown that when we knock down these mRNA methylation enzymes and combine with the target therapy, BRAF inhibitors, in fact, increase the sensitivity of the melanoma cells to the target therapy. And more interestingly is that actually when we combine translational a better response. I think there's somebody didn't.
The voice is lagging.
Yeah, the micro. I heard an echo. So with this translation inhibitors, actually we observe a much durable response to the target therapy in our melanoma xenograft mouse model. So collectively, actually, raise the concept that in the melanoma methylation of the mRNAs on their 5' UTR or the 3' UTR nucleation of the mRNA, they have this selective translation activity on different mRNA populations, which may be involved in the persistence or the deceleration of their growth rate. But in the parental cells, since we have enough ribosomes and most of the mRNAs, they can be translated into proteins. So those mRNAs that are more involved in the proliferation, and the cells at the same time are sensitive to our target therapy treatment. So based on these works, that we were wondering how the persistent cancer cells, within the persistent cancer cells, how the ribosomes can selectively translate different population of cells, if it's only based on the methylation or it's due to other factors. So one thing is that, in fact, we observed a dramatic reduction of ribosome level in the we observed a reduction of the ribosome level in the persistent cancer cells. As you can see here, there's a immunofluorescent staining of the ribosome biogenesis center of the nucleolus in the nuclear. And so we think that we have a hypothesis that in persistent cancer cells, maybe the reduction of the ribosome create a kind of like a bottleneck for the translation of the mRNAs in the persistent melanoma cells. And this bottleneck may further promote the cells to selectively translate different population of mRNAs at the same time to survive some kind of cellular stress or nutrient deprivation. So these observations show that actually we were thinking this is different from the genetic mutational resistance that most of the laboratories are working on. So for the genetic resistance, actually some kind of a Darwinism evolution theory. So it's like the treatment is like a selection of those cells that already have a priori genetic mutations, heterogeneity, and only those cells with the resistant related mutations, they can survive after target therapy treatment. But with the persistent cancer cells, actually, we were proposing another scenario is that the Lamarckism plus Darwinism evolution theory, we think is happening at the same time in the population of melanoma cells. It means that not only those cells a priori have resistant related mutations can survive, but also those cells that they can switch their phenotype without additional mutations upon the treatment of BRAF and MEK inhibitors, they can further survive this treatment selection. So the most important factor in this kind of intrinsic adaptation to the target therapy is that the interaction between the tumor cells and the treatment. So the tumor cells, we think that they should have at least three very important features that they can adapt to these stress-induced treatment conditions. At the same time, their phenotype switching is reversible, as we observed before, for the translational activity and also their sensitivity to the same drugs. And at the same time, the optimality is determined by the genetic background of the melanoma cells. So it doesn't mean that they can change to anything they want. So as we can see that before, I showed that they have this kind of 5-cell state based on the single-cell RNA sequencing. So their phenotype actually is predetermined by their genetic backgrounds. So how they can survive this kind of target therapy is largely determined by the genetic mutational background of the melanoma cells. And this actually is applicable to the immunotherapy anti-PD-1 inhibitors as extrinsic adaptation. So the idea here is also a very important interaction, not only between the melanoma cells and anti-PD-1 antibodies, but also the most important factor is the immune cells, how these three factors interact with each other to promote the immunotherapy-induced persistent cancer cells is one of the direction that my lab is continuing working on. So in 2020, actually, we wrote a review that to try to summarize all the studies that have been done on the persistent cancer cells. So as you can see here, the persistent cancer cells nowadays that they have 4 major mechanisms to help them to survive, no matter it's chemotherapy or target therapy, or even immunotherapy. So in general, they will have since they do not have secondary mutations, they will have this kind of epigenetic alterations and also adaptive mutabilities that to slow down their proliferation, to survive this kind of cell stress, and also adapt to their cell metabolism from the ribosome dependency point of view, and also mitochondrial respiration and oxidative trait of some of the persistent cancer cells have been shown are very sensitive to the ferroptosis. At the same time, in different type of cancers, the cells, as they can change their phenotype. At the same time, they can switch their cell identity. For example, in lung cancer, some researchers have also observed that upon the treatment of target therapy EGFR, the adenocarcinoma lung cancer, they can transdifferentiate into small cell lung cancer. So which is kind of also a adaptive phenotype switching upon the treatment. And also most importantly is how they can hijack in the microenvironment of the tumor immune system to evade the immune system during such a long time of remission in the patient bodies, and they can relapse after, like, for some of the patients, they can relapse even after 7 to 10 years. So how they can stay hide it from the immune system during such a long time is also a very important factor that are supporting these kind of persistent cancer cells to survive the anti-cancer therapies. So I will stop here. I would like to thank the collaborators in France and also in China. And I put several pictures from my province in the west of China. I hope everybody will have some chance in the future to come here to visit. Here is a really nice place. Thank you very much.
Well, thank you very much, Shensi. It was a really, really wonderful presentation. And before giving avant de donner la parole à Kamel, actually I have some basic question regarding this. Because I was working for about 4 years in Sweden, and melanoma was a main problem there. And I want to ask you, why working on melanoma actually changed? Why this kind of cancer, and why this kind of cells? As you can see, it's a really general question. And then, to integration, you showed in a certain moment that the relapse was mainly on the same site, in the beginning of your presentation. And I'm really curious to know and to ask you if it's something related really to melanoma, or you can see, or we can see this kind of same site relapse within other cancer. And the story of switching the phenotype without any genetic change. So again, is it really something specific to your cells, or we can see this phenotype more universally? Voilà, thank you.
Why? Well, it was stunning that you said in Sweden they also have this kind of melanoma problem, because I think they exposed to the sun much less than the French or easy. And in the world, the most I think it's Australia. They have the most serious problem of the.
Yeah, New Zealand, they have really problem. I mean, it's a real problem there, but also in the Nordic countries.
So why I want to work on the melanoma, actually, well, it's a main problem. When I was working in France, they are increasingly will have more than 7,000 cases every year of melanoma. And I think during in the 1980s, the people, when they go to the beach, they do not actually, at that time, they don't protect from the sun. So now the time that those people from that ages, they start have this kind of develop the different melanoma. And on the other hand, I think melanoma, as you said before, it's really a pioneer now in the cancer domain, that especially for the immunotherapy. Most of the anti-PD-1, for example, and the anti-CTLA-4 was firstly approved in a melanoma treatment. So a lot of nowadays, a lot of clinical trials, for example, combination of the anti-PD-1 with other immune stimulators like TLR9 and also oncolytic viruses were first done in melanoma. So I think it's a very good model to study not only to the target therapy that I talked today, but also the interaction between the tumor cells and immune cells how they respond to the therapy, I think is the best model. So that's why I was really interested into melanoma work. And for the relapse, yeah, it's not only the melanoma patient the problem of relapse. Of course, it's for example in China now. I also worked on the lung cancer since luckily in China we do not have a lot of melanoma patients. They from the sun, and so it's the same for the lung adenocarcinoma or the small cell lung cancer is the same. Actually, for example, lung adenocarcinoma, they have this target therapy for the EGFR mutation, and now we have also the KRAS mutation. The KRAS inhibitors was approved recently. So the problem is also the relapse was very quick. Actually, for the lung cancer is less than 6 months. So I think they have also for the breast cancer and also like the ovarian cancer for the chemotherapy. In fact, we have very similar problems. So I think it's not only specific to the melanoma for sure. And for the phenotype switching, it's the same. Well, I cannot say that that's the, for example, the translational regulation, whether it's a general phenotype that we can also observe in other type of cancers. So I cannot say that because this is really specific to the melanoma. And also the metabolism, I think, is specific to the melanoma. Whether other type of cancers, they also have this kind of fatty acid oxidation involved in their survival, is I don't think so. And there are some studies, for example, in lung cancer. In fact, for the metabolism part, they are more dependent on the glutamine metabolism rather than the glycolysis. So I think it's different because the initial genetic background is totally different compared if we are comparing melanoma to lung cancer. So this kind of landscape is totally different between different type of cancers, and the phenotype switching, I think they have this phenotype switch, but maybe the underlying biological mechanisms are totally different.
Yeah.
So it's difficult to generalize. I mean, the same mechanism to all the other type of cancers.
Yeah. Well, merci. So if anyone, I mean, Kamel, please go ahead and.
Well, terrific presentation and the terrific work. Actually, we are really impressed. And I have a question. Actually, several naive questions. What about the non-responder to the targeted therapy? What about the genetic backgrounds? You know, it's like you looked at you focused on the one who you focused on the cells that do respond to treatment and not. But what about, you know, it's like the population of responder versus non-responders? Like is there a genetic predisposition to not responding? Is there polymorphism that are involved in the fact that you do not respond to the treatment?
Yeah, in fact, those patients, they are primary resistant patients. So this is largely due to the heterogeneity of the tumors. Of course, those cells do not respond initially. They harbor other resistant associated mutations. So this is totally another scenario. Okay. And as I told before, in melanoma, in fact, for the target therapy, if they harbor the BRAF mutation, their response rate is really high. It's almost to 90%. So 10% patients, they do not respond. It's true that they also have some other mutations that promote their survival.
Okay.
And I want to, since you were asking this, so actually, as I showed before the 2014, in fact, it's a monotherapy. It's only BRAF inhibitors for the treatment of melanoma, and we get in resistance very rapidly after 4 to 6 months. And there is a phenomenon in the melanoma. It's called paradoxical activation. So it means that the BRAF inhibitor we were treating the patient, they can activate, not inhibit, they can activate the wild type BRAF proteins. So there are a lot of wild type BRAF proteins in the immune cells, and they also in the bad immune cells like T regulatory cells and also MDSC cells. So that's why since 2014, the first line treatment, we do not use any more monotherapy. We always combine the MEK inhibitor because it's downstream of the BRAF. So that's why now we do not have this paradoxical activation of BRAF.
Okay.
And the advantage of this is they have less toxicity actually compared to the monotherapy because they inhibit the wild type activation. Yeah.
Okay. Okay. And what about the immunotherapy? As like the PD-1, so it's like you combine, so in the treatment, there is a combination of immunotherapy and targeted therapy or not right now? And it's like, what's the actual treatment?
Now the first line treatment in melanoma, in fact, is immunotherapy. So when a patient came to the hospital, we do not sequence their tumor cells. We use the anti-PD-1 immediately. If they don't respond, then we will look at whether they have the BRAF mutation, and we switch to target therapy.
Okay.
In clinics, we're already using the combination. We're using the combination, but the problem is the toxicity. As you may know that the anti-PD-1, they have less toxicity compared to CTLA-4, but when we combine with BRAF and MEK, the grade 3 to 4 toxicity is high to 20%. So it's very toxic. So for this, actually, it's using in clinics, but should be very careful.
Okay. I have I was very puzzled about you know, it's like what related to what you're doing is about the fatty acid oxidation pathway. It's very interesting. And is there you talked about fatty acid inhibition pathway? Is it possibly could it be possibly used in the future for therapeutical purposes? Or and if I may say, you know, it's like based on what you're doing, why you know, it's like it rings a bell to me because we work on 3 types of population. When I'm not getting into details, but immune population, and some of these population use the glycolysis pathway, and others use the fatty acid oxidation pathway, and it's mTORC2, you know, it's like related. It's the Richter instead of the Raptor. So you talked about Saturn. Is it possible to envisage Saturn treatments in the melanoma situation?
Yeah, it's a good question. We were working on this. Since the inhibitor we were using actually is FDA-approved drugs in the 1980s, treating for the psychiatric diseases. But the tiohidazine drug was removed by the FDA since 2005 due to cardiac disease, very severe diseases. So they were removed from the FDA. But I think for the combination treatment, it's possible because if we combine, especially if we combine with the target therapy, the concentration we were using is much lower than the fatty acid oxidation inhibitor used as a monotherapy. This is what we are trying to further work, especially in vivo and also some because the model we were using actually is a nude mice, so they do not have immune system. That's the big problem. Because as you said that fatty acid oxidation, we know they are participating in the regulation of a lot of immune cells. So if we inhibit this, whether we will have a large effect on the systemic level.
It's true. It's true. It's true. You're right. But you know, it's like not, you know, in a differential way. There, I mean, it's like we are surprised. You know, it's like how some of our subpopulation rely more on oxidation, which is why I'm telling you mTORC2, because you can leave other immune system working and taking off other, you know, it's like I'm talking about NKT17 versus NKT2 and NKT1, the TH1, TH2 versus TH17. But anyways, like, yeah, it's you know, preferentially immune system cells, also innate at least, use differentially these drugs. So you can you know, break down your some pathway and used by some cells and leave other cells working, you know, it's like, but yeah, it's I get your answer actually.
Any other
yeah,
question,
Mohammed.
maybe? Any other question from the audience? Aziz, yes, please go ahead.
Thank you, Mohammed. Thank you, Dr. Shen, for this very nice presentation. I enjoyed it, although I'm not from the domain, but it was a very important presentation and actually very informative. And I don't have a question because, as I said, I'm not from the domain, but here, just when I've seen your presentation, I don't know either what I'm gonna say is correct or wrong. You can correct me later on. I think that here it's okay dealing with tumor cells, with melanoma, or with these populations. I guess that it's kind of dealing with the community. It's we need here to let's say classify these cells or these tumors or whatever as a community, because from your discussion or from your presentation, we can see that at the beginning it's okay. We do have here a response to this kind of therapies, but later on there is like a resistance. And this is just, I'm thinking loudly. It's like, it's okay, we do have this community of cells that are not, that are rejected by our body by taking this kind of therapies. And then it will be like a war, let's say, between them and these molecules that we are rejecting. Meanwhile, it's okay, we'll have here like this community, they will sacrifice with their cells, and meanwhile, there will be like signals between the defense and other type of cells that will here later on take this persistence, or what we said here, resistance. So I don't know, because here, one of the examples you showed that it was, the tumor was there, and then after 15 weeks or 15 days, I don't remember, the phenotype becomes normal, and then we'll have again the abnormality. So is there any possibility, at least if what I'm saying is correct, during the therapy to go and find a way to kill the future, let's say, resistant cells? And as I said, maybe these resistant cells, they are using the information coming from the other cells to develop such type of resistance, maybe by modifying the epigenetic factors, maybe by modifying the DNA. I don't know what happened exactly, because as I said, I'm not here from the domain, but I'm thinking just loudly, and I want to get an explanation from you as expert. And thank you again.
Yeah, it's a really important question. And as I said before, most of the observations we have seen, it's in vitro. And I think most of the labs working on this domain, the challenge is that we do not have a very good in vivo models. And we think it's not only the intrinsic factors are determining the persistent phenotype. Of course, not only other cells, they can send messages to this population, and also this population, they can also send messages to other cells in the environment. And I remember there was a paper in, I think it was in 2018 in cancer cell. They also observed how the matrix of the tumor bed during the target therapy treatment, how they remodel their matrix to help the cancer cells to survive without additional mutation in the cancer cells. So of course there is a intense communication between the cells within this community. It's true, it's a whole ecosystem. That's why what we are doing is trying to connect some patient samples, and by using single cell techniques, not only look at the melanoma cells, but also the other type of cells, including the fibroblasts, the immune cells, and even the endothelial cells, how they communicate with each other, and how the tumor cells engage other normal cells to help themselves to survive, and how they change their phenotype upon the other cells, how they modify the environment. This is a very important question, an important direction that we will continue to work in the future for sure.
Well, so again, thank you, Aziz, for the wonderful question. Shensi, it was fantastic. I'm very happy first to see you again after all of these years, and I wish you all the best for your adventure in China.